Hello everyone, myself Professor A.S. Mochi, Department of Agriculture Entomology, Wagh College of Agriculture, Nashik. Today we are going to learn about the microbial control of the pest management, or microbial control is nothing but the one of the we can say that the applied branch of the biological control. But here in that microbial control we can use a different microorganisms or the insect pathogens in the pest management. Let us start. What is meant by microbial control? The definition is that it is the branch of biological control which deals with the study and the utilization of the microorganism for the suppression of the pest population density. So the question is that which are the different microorganisms or insect pathogen we can use in the microbial control? Here the various fungus, bacteria, virus, protozoa, rickettsia, and we can also use in a or as well as the entomopathogenic nematodes are the different microorganisms we can use in the microbial control. Here, the one of the most important scientists it is related to the microbial control, that is the E.S. Tenhorst. The first time give the term microbial control, and is he was referred as the father of insect pathology or father of microbial control. Then, so we can use the different microorganisms or pathogens for the suppression of the pest population in the agroecosystem. So, question is that how or which are the different ideal characteristics, or which are the different characteristics for ideal insect pathogens? These are the some of the qualities or characteristics for the ideal insect pathogens. It should be host specific, it should be virulence, it should be toxin production, a rapid spreading of the disease, persistent for long time, then amenability to mass culture, cost effective and economical, safe to non-target organisms. These are the some of the general qualities the insect pathogen should have these qualities. So let us start. Which are the different microorganisms? In that, the first and most important microorganism is that fungus, and here that fungus is called as entomopathogenic fungus. It means that the fungus which cause a disease in insects is called as the entomopathogenic fungus, also called as insect fungus. The fungi that can act as parasites of insects and kill or severely disable them. Okay, then these are the some of the symptoms of fungal infection. When the fungus is attack on the insects or insect body, which are the different symptoms which is produced by the insects during their infection? These are the general symptoms that is the loss of appetites, irritability, paralysis, discolor patches on the integuments like that. Okay, and death occurs after the development of the fungal infection inside the body of the insects. The death occurs of that particular insect in near about 4 to 7 days depending upon the host insect and the environmental conditions. Then these are the some of the entomopathogenic fungus, that is the Metarhizium ansopli, Beauveria bassiana, Verticillium lecani, Nomura rilli, Hirsutella thompsoni. It is very effective against the different insect pest populations. The Metarhizium ansopli, it is called as a green muscady fungus, and it is very effective against the coleopteran insects. Beauveria bassiana, it is called as a white muscady fungus, effective against the lepidopteran insects. Verticillium lecani, effective against all the sucking pests. Now this Verticillium lecani is also called as Lecanicillium lecani, also called as white halo fungus. Nomura rilli, it is very effective against nowadays against all types of caterpillars. Then Hirsutella thompsoni, it is one of the most important entomopathogenic fungus against the mites or phytophagous mites. Then now how or what is the mode of actions of entomopathogenic fungus in the field? When you spray this entomopathogenic fungus in the infested fields, and this when the insect it is comes contact with this all types of fungus, the first step is that the adhesion of spores to the host cuticles. It means that this fungus is attack on their cuticles, or it is attached to that cuticles. Then after creating the favorable condition, the spore germination is takes place. Then after spore germination, the penetration of the cuticle is occurs. The spores is penetrate into the body. Then the growth in the hemocoels. It means that after the penetration of the spores into the that midgut, or we can say that the hemocoels, the sporulations or the growth of the fungus, it is takes place inside the hemocoels of that insects. Then near about 5 to 6 days after the 5 to 6 days, the death of the insect is occurs and due to the saprophytic feeding of that fungus. And last is the hypal re-emergence and sporulation is occurs, and then this spores is against searching their another host. Then this is the field application of or the dose of the field application of different entomopathogenic fungus against the different pest. The next pathogens is that insect virus or entomopathogenic virus. The virus, it is coming under the family Baculoviridae and cause a disease in the Lepidopteran larvae. So it means that the virus it is mostly effective against the larva. There are the mainly two types of virus. The first one is the NPV virus and second is GV virus. In that NPV, the nuclear polyhedrosis viruses HANPV, SLNP and GV are the most important. Sorry, and in case of GV, the granulosis virus, the CIGV is most important. HANPV means Helicoverpa armigera nuclear polyhedrosis virus. It is very effective against the American bolome. Then SLNPV, it means that Sporoptera lutura nuclear polyhedrosis virus effective against the tobacco leaf eating caterpillar. Then CIGV, it means that Chiloinfuscatellus granulosis virus. It is very effective against the early shoot borer of the sugarcane. And this is the mode of action of the entomopathogenic viruses. After the virus is attacks or virus is it is we can say the ingest by the particular caterpillars, and the virus it is enter into the midgut midgut of that insects, and it is we can say that the replication of the virus is occurs inside the host or hemocoels and causing or forming a occluded virus or occluded bodies, and insect it will be killed. Then here these are the some of the symptoms which is produced due to the infection of the virus. Here the Lepidoptera larvae, it became a sluggish, pinkish in color and lost their appetites. Then body becomes fragile and ruptures to release a polyhedra or virus occlusion bodies. Then most important is that the dead larva hang from top of plant with prolegs attached. That symptom is called as tree top disease, and this tree top disease is nothing but the one of the typical symptoms produced due to the infection of the entomopathogenic virus in insects. Then these are the some of the virus HNPV, SLNPV, MANPV, ANPV and GV. It is very effective against the particular pest, and this is the field application dose. Then next is the next insect pathogen is insect bacteria or entomopathogenic bacteria. This is nothing but the one of the simple classification of the entomopathogenic bacteria. The bacteria is classified into two major categories: spore forming bacteria and non-spore forming bacteria. In spore forming bacteria, the obligate and facultative bacteria, and in case of non-spore forming, the Serratia is one of the most important bacteria. Then here the spore forming obligated bacteria, Bacillus papilli and Bacillus japonica, it is very effective against the root grub or white grub. It is nothing but the spore forming obligatory bacteria. Then in case of facultative spore forming, it is nothing but the Crystalliferous, that is the BT, and the BT is very effective against the Helicoverpa armigera and the Subodoptera lutura. And in case of facultative non-Crystalliferous, the Bacillus cereus, it is very effective against the Hymenopteran and Coleopteran insects. Then these are the mostly used bacteria in the pest management, that is the Bacillus thuringiensis, Bacillus cereus and Pseudomonas fluorescens. And after the infection of the bacteria, these are the some of the symptoms which is produced due to the infection of the bacteria. Then here the spore forming bacteria, that is the facultative Crystalliferous, nothing but the BT. That BT produces spores. That spores is called as, or toxins is called as endotoxins, and this endotoxins, it is paralyzes the gut when ingested by the insects. Okay, and it is very effective against the Lepidoptera insects and is nothing but the stomach poison. It means that the bacteria are the BT are the stomach poisons. Then in case of spore forming, the Bacillus papilli, it is very effective against the coleopteran grubs or coleopteran insects, especially for white grub, and it causes a milky disease in the grubs. Then in case of non-spore forming, the Serratia, it is very effective against also the grub. The next is entomopathogenic nematodes. Here the entomopathogenic nematodes, it means that it is nothing but the beneficial nematodes. The nematodes which are capable of killing, sterilizing, or seriously hampering the development of insects and completing at least one stage of their life cycle in the host. Here, the most important groups of the EPNs or entomopathogenic fungus are the Stenorenema and Heterorhabditis are the most important groups. These are the some of the special qualities of the standard nematode and heteronematode EPNs, and it is very effective against the different insect pest population. Here, the symbiotic association with the bacteria, that is Xenorhabditis and Stenonematode, and the Photorhabditis and the Heterorhabditis, it is nothing but one of the most important bacterial. The infective stages, that is the third stage, juvenile, is a non-feeding, free-living, durable, and capable withstanding the adverse climatic changes or conditions. These are some of the most important characteristics. Then this is the mode of action of EPN. When the EPN it is attack on the insects, the J3 infective juvenile enter the blood system of the host through the opening, that is nothing but the cuticles, and after the entering the juveniles of that EPNs, the symbiotic bacteria released by the nematodes, and with the help of the symbiotic bacteria, the we can say that the we can say that the multiplication is occurs. Then after the first and second generation nematode develop on the dead host, and after that infective juvenile it is leave the host, and then another next is that they searching their new host. And these are the some of the advantages of the microbial agents as a component in the IPM. Then these are the some of the factors which is affect on the biological control. The tolerance limit of the crop to insect injury, crop value, crop duration, indigenous or exotic pest. Then if alternate host available for natural enemies, control target pest is less. Okay, so these are the some of the important factors which directly affect on the biological control. Then these are the some of the limitations is also for the biological control. Thank you.